The chapter called as cellular respiration, and we have started the first part. From the discussion that we had in the last class, what we remember, respiration ki hoy, it is a reaction, and whenever we talk about reaction, what type of a reaction is something that we have discussed, je anabolic reaction, ne catabolic reaction, endergonic, ne exergonic, oxidative, ne reductive, we can discuss koriso. Tarpor what we said is, we have also used certain words called as complete oxidation and incomplete oxidation, right? Types of respiration, protoplasmic respiration and floating respiration, aerobic respiration and anaerobic respiration. Last class what we complete koriso, we have completed the section called as fermentation. Fermentation or anaerobic respiration, obviously it's the same, but one within the body of an organism, one outside the body is generally how we define both of them. Alcoholic fermentation, lactic acid fermentation we have done, and what is the difference, what is the similarity, hekini ami koriso, right? So aji ki korim, we will be talking about the mechanism of respiration, and talking about mechanism, obviously we will be talking about in details about the aerobic respiration, right? Before starting, just to remember from the point that we have discussed in the last class, respiration boli kole what we understand, we have used the word called as respiratory substrate. A respiratory substrate mane ki, the food that gets oxidized, and as a result what gets synthesized is ATP, carbon dioxide or water. No? Complete oxidation, incomplete oxidation or meaning to ki asile? If what? Respiratory substrate gets oxidized and ultimately they form ki carbon dioxide, water and energy, gutei tinta jedi ulai tetial tak ami ki boli kom complete oxidation, jedi nulai tetial tak ami ki boli kom incomplete oxidation, right? Aerobic respiration boli khwa loge loge we understand which process, it's obviously a complete oxidation process, right? Next is talking about respiration, the steps of respiration. Steps boli kole respiration has been defined or discussed under 3 steps. The first step being glycolysis. Glycolysis or pisot ki hobo Krebs cycle or pisot the third step is terminal oxidation, right? So aji ki korim glycolysis or arambho korim. So heading koriba mechanism of respiration. Kya apan point likhi lo, tar pisot we will discuss glycolysis on the first step. So talking about the first step or the initial step boli kole what we are talking about is glycolysis, no? And last class to we discussed koriso, we have just started, no? Eta line ami ki kosilo, so whenever we are talking about glycolysis, what we understand, breaking down of the glucose molecule inside the cell with the help of oxygen, right? And obviously different enzymes that are associated. Our glucose eta bhangi pelai cell to bhitor ki form kore, ultimately at the end of glycolysis we see the formation of pyruvic acid, right? Next is glycolysis junto first step hoy dependent of oxygen, independent of oxygen. What we have said is oxygen plays no role in this process. Aerobic respiration or first step ki hobo glycolysis hobo, anaerobic respiration or first step ki hobo glycolysis hobo. It's only after pyruvic acid that will decide say aerobic hobo ne anaerobic hobo. Pyruvic acid is jeti oxygen loi pelai oxidized hoy into further products, tetiale hetu process ki hobo aerobic respiration, and if suppose pyruvic acid gets further broken down in absence of oxygen, tetiale tak ami ki boli kom anaerobic respiration. So I think this line is understood, right? ইয়াৰ তলত এটা বস্তু লিখি লোৱাচোন। ইয়াৰ তলত লিখিবা হা
Incomplete oxidation of C2H10OH.
I think this is understood, right? Anaerobic respiration বুলি কৈছো, but ইয়াতে anaerobic respiration তো carbon dioxide form হ'ব fermentation process to, right? কিন্তু ইয়াতো ঠিক তেনেকে last class যেনেকে discuss কৰিছো, looking at this, যদি আমি fermentation point of view পৰা কথা কৈছো, then we are talking about which type? Alcoholic, right? কিন্তু in normal, alcoholic fermentation is the most common one, so we depict it by this only, right? Okay, next is talking about the major pathways of respiration. Duta pathway ase, PPP, but common pathway, right? Normally, whenever we talk about the common method of respiration or breaking down of food or oxidation of food, generally we talk about this. Kintu ketiyaba according to certain plants, according to certain cells, tahator they have an alternated method of glucose breakdown. সদায় যে অকল এই তিনটা ষ্টেপে হ'ব, কমনলি ইন মোষ্ট অফ দ্য চেলছ কি হ'ব, গ্লাইকোলাইসিস, ট্ৰেপ সাইকেল আৰু টাৰ্মিনেল অক্সিডেশ্যন হ'ব। কিন্তু কেতিয়াবা কি হয়, কিছুমান প্লান্টছৰ কিছুমান চেলছত অল্টাৰনেট ৱে ইন আনাদাৰ মেথড কি হয়, গ্লুকোজ টু ব্ৰেক ডাউন হয়। চ' দ্যাট মেথড ইজ কল্ড এজ পেণ্টোজ phosphate pathway. We'll be discussing this also, what is the significance, etc., etc. Ketia hobo. We'll be talking about it later. Kintu respiration boli kole commonly discussed kuntu obviously the first one. Right? Why is it called as common pathway? Because it's most commonly seen. Okay. Ekhini jodi hoy gol glycolysis heading koriba ebar. Last class of glycolysis to likhibo disu, likhisila no? Oh, baki june likha na tane likhi loba aji. Ekhini hol no? Kiba doubts asile niki? Ekhini loke clear? Okay. So, ever likha glycolysis. Glycolysis or ketaman points. Ami first likhi lo and okay. Glycolysis or ketaman points likhi lo. Important points, right? And then we'll write the 10 steps of glycolysis. all the points are important, eh? Right, ekhini points important hai hoy gotekhini, right? Who discovered it, right? Emden, Meyerhoff and Perennis, and so it is also called as the EMP pathway. So glycolysis process to quote hoy, you have to be precise because we know respiration consists of 3 steps and we have to know where exactly the 3 steps takes place. Krebs cycle quote hoy, glycolysis quote hoy, terminal oxidation quote hoy, right? So glycolysis quote hobo cytoplasm. So all the products that are synthesized because of glycolysis could produce hobo in the cytoplasm itself, right? Next is, since we know that it's a series of reaction, kiman step reaction, 10 step reaction, and protective step or conversion or ki hoy, there are certain enzymes that helps in the process. Aru এই enzymes bilakor cofactor hisabe eta mineral thake, that is Mg, right? So Mg report question ahe kuntu mineral generally help kore. Next, glycolysis, as we have already talked about, no, glycolysis no doubt is the first step of respiration. Respiration boli kole what we understand, taking in of oxygen and giving out of carbon dioxide, but in spite of that glycolysis is a step where oxygen taken no kore, consume no kore, but carbon dioxide evolve no kore. Generally when we talk about evolution of carbon dioxide, oxygen to consume bilkule komanu no kore date, but if you are talking about evolution of carbon dioxide in the 10 step reaction, there is one particular reaction jotnike one molecule of carbon dioxide gets liberated. Ami etia olop pisot reaction khini likhim, no, the 10 steps will write, and we will see that in one step eta reaction on ki hoy, one molecule of carbon dioxide gets liberated, but we know respiration on okol eta molecule is not, I mean it's not enough, right? Pratek bare oxygen goes in, carbon dioxide ulaya hai, so we cannot be saying that it's just one molecule of carbon dioxide comes out. So generally ami kuntu process out of the 3 process, juntu process hote ba out of the 3 steps, juntu step hote maximum carbon dioxide ulai, he step to khe ami assign koru as the step because of which carbon dioxide gets liberated. And what we are talking about is exactly Krebs cycle, right? Out of the 3 products, no, as we have already mentioned, 3 products boli kole ki hobo laibo, energy hobo laibo, carbon dioxide hobo laibo aru water hobo laibo. So carbon dioxide evolution, this credit goes to which step? It goes to Krebs cycle, karon Krebs cycle ot 4ta carbon dioxide ulai, right? Yate khali eta ulai, so we don't consider, right? Next is, as far as water is concerned, water we'll study later. So water kuntu step or karne hoy boli kwa kole, glycolysis to eta step out we will see the evolution of, the release of water. Kintu jihetu eta molecule, we don't give the credit to glycolysis. So questions idea hai, where exactly is or which process is responsible for the synthesis of water in respiration? It is terminal oxidation, right? Thik teneke carbon dioxide kon glycolysis to ulai matra eta molecule ulai, hekane ami neglect kori diu tak, right? Kintu kuntu associated hobo, Krebs cycle is associated hobo. But this line is important. Question to jodi ahe, eta molecule ulaise, but in spite of that, since we don't consider, so this is a line that you have to remember. Glycolysis is a step jotnike oxygen consume no hoy, carbon dioxide evolve no hoy. এটা হয় হেকাৰণে আমি তাক নেগলেক্ট কৰো okay anyway next is what happens in the process one glucose gets broken down into pyruvic acid eta glucose bhangile keita pyruvic acid form koribo two molecules of pyruvic acid they will form pyruvic acid is a how many carbon compound it's a 3 carbon compound we have studied about something like 4 carbon compound 3 carbon compound pga phosphoglyceric acid porhisilu no oxaloacetic acid porhisu malic acid porhisu eblak 4 carbon 3 carbon pyruvic acid ki hoy 3 carbon compound next is during this steps ki liberate hoy NADH2 aru ATP form kore ATP to form koriboi lagibo no because that's a form of energy aru tar logote reducing cofactor that is NADH2 form kore NADH2 ultimately again breaks down and forms ATP itself right so ultimately glycolysis or 10 step process of ki synthesized hoy out of the 3 products that we have talked about it is only energy in the form of ATP right carbon dioxide hoy niki no hoy water hoy niki no hoy kintu okol ATP hoy ATP o ako duta weight hoy eta hol we use a word called as substrate level phosphorylation if you have gone through the process no substrate level mane directly glucose to bhangi pelai keita ATP ulaise that is what is called as substrate level aru eta step out ki hoy the ATP gets generated via NADH2 what I said is NADH2 aru ATP produce hoy right NADH2 ultimately break down kori pelai ki form koribo ATP e form koribo so directly keita form korise aru NADH2 bhangi pelai keita form korise directly to form korise tar ami eta term use koru that is called as substrate level phosphorylation phosphorylation to what we have already explained no it's a formation of ATP from ADP that is called as phosphorylation so within the 10 step reaction directly keita ATP formation dekha pam aru NADH2 bhangi pelai keita form koribo and as we have said before NADH2 eta molecule keita ATP synthesize kore tinta so dutai keita koribo obviously sota so glycolysis of ATP synthesis kiman hoy 8 ATP out of that direct keita hoy duta aru baki sota ki hobo NADH2 bhangi pelai hobo and so we have written the word called as via ETS mane electron transport system NADH2 to bhangi pelai electrons protons separate hoi pelai tahate eta pathway jabo or ultimately ATP will get synthesized right what we are talking about is the third step of respiration that is terminal oxidation process of ki hoy NADH2 bhangi pelai electrons or protons bilake it plays a role later we'll study is this term understood substrate level understood so question directly keneke ahibo pare either it will be asked the glycolysis of ATP synthesis kiman hoy then we'll be talking about it as 8 or question to ahibo pare substrate level phosphorylation glycolysis kiman hoy 2 right or total kiman hoy total hobo 8 substrate level hobo 2 via ETS jodi question ahe via ETS kiman hobo 6 or through or via NADH2 boli jodi word to ahe tetio ki hobo 6 right so teneke ami bostu to pohilom so these were the information now we'll write the reactions এখন ডাঙৰকে পেজ লবা হা ৰিঅ্যাকশন লিখিবলে Z6P বুঝি পাও ন গ্লুকোজ 6 ফসফেট Hmm. Single. Second to both way. Reversible, irreversible to Sabah. Huh? Or 4, a bracket of IP, inorganic phosphate. কোনকেইটা স্টেপত ATP প্ৰডিউস হৈছে আৰু কোনকেইটাত NADH2 প্ৰডিউস হৈছে সেইকেইটা স্টেপ বেছি ইম্পৰ্টেন্ট ঠিক আছে ইফ ইউ সি ইন দ্য ফাৰ্স্ট এণ্ড দ্য থাৰ্ড ৰিয়াকশন তাত ATP প্ৰডিউস হৈছে নে কনজিউম হৈছে ATP কনজিউম হৈছে সো প্ৰথমতে গ্লাইকোলাইসিস প্ৰসেসটো যেতিয়া স্টাৰ্ট হয় ফাৰ্স্টতে ATP production নহয় they start the process by taking in ATP from the cell itself right with the help of enzymes gradually break down hoy and in the process they will synthesize ATP as far as likha hol likhin nai hono likhilwa If you are done, please say. Okay. Reversible, irreversible kita saba kun kitat first, third or last, right? Baki soap reversible, first, third or last irreversible. Hekhini ami likhi dim points, hekhini upor to questions ahe. We'll be writing it down. Okay, finished? Oh. Huh? 11 step in the first step to consider korani. It's just a starting too. Eleven বুলি ক'ব লাগিলে actually to 10 ন তুমি গ্লুকোজ টু পৰা যদি ষ্টাৰ্ট কৰা এইটো আৰু এইটোতো একেই ন বুজি পাইছা ইয়াতে মই এটা জেগা নহ'লে কাৰণে ইয়াতে প্ৰডাক্টটো লিখা নাই তো ইয়াতে কি হ'ব তোমাৰ এইটো ৰাইট সো গ্লুকোজৰ পৰা তুমি তেনেকৈ কৰা কি সুধিছিলা ডাইফসফোগ্লিচাৰলডিহাইড ডাইফসফো ফুলকে লিখিলেও লিখিবা 1,3 ডাইফসফো দুটা ফসফেট আছে ন ফাৰ্স্ট পজিশন আৰু থাৰ্ড পজিশনত সো 1,3 ডাইফসফোগ্লিচাৰলডিহাইড কি হলে কনভার্ট হৈছে গ্লিসারিক অ্যাসিড হলে কনভার্ট হৈছে হয় হল Okay, so talking about these steps, no, sabade ei bostu tu kintu yate ako belekke start hua nai. It is the continuation, no, transphosphorylase or pisot ki form korise 3-phosphoglyceric acid, right? Okay, starting glycolysis, let's talk about this. What we have said is glycolysis is the process in which the glucose molecule gets broken down and one glucose molecule gets broken down to ultimately forms kiman 2 molecules of pyruvic acid. So pyruvic acid keta form kore duta form kore. Next, enzyme mediated process to protect to step out different enzymes there are associated and plus enzymes to ase tar report a certain types of minerals. Which one is the most important? Obviously Mg2 positive, right? Hebilake cofactor hisabe kam kore. Next is so first is you have to by heart the steps protective reaction ami mon rakhibo libo. As far as the enzymes are concerned, no, if you see in your NCERT book gute gita enzymes dia nai kintu, right? Ketaman he enzymes dise. So as far as these enzymes are concerned, there are just 4 enzymes that we remember and it's important from examination point of view. So board's point of view pra ki koribo glycolysis process to likhibole dibo. Entrance point of view pra to glycolysis process to likhibole ni diye. Question to keneke ahibo pare je aldehyde to acid by aldehyde to acid kune form korise phosphoenol pyruvic acid or pyruvic acid kuntu enzyme involved ki produce hoise teneke step wise question ahibo. So from entrance point of view, there are 4 enzymes that are very important. First, obviously the first one, hexokinase. No, hexokinase to kami ki boli kon the starting enzyme. No, jehetu process to kune start kore hexokinase. So hexokinase starting enzyme, it is important. Next is come to aldolase. Aldolase is also called as splitting enzyme. Why is it called a splitting enzyme? Because we have seen fructose 1,6 diphosphate dubhagot bhag hoy goise, right? So split koribole kune help korise aldolase. So first, second, next is enolase. Within this process, as we have discussed, carbon dioxide evolution, oxygen role, carbon dioxide role eko dekha nai, kintu at one point ki hoise water evolve hoise, kintu just one molecule. Jehetu enolase is responsible. Enolase is called as a dehydrating enzyme. Question ahibo pare, name the dehydrating enzyme associated with the process of glycolysis. It is enolase. So enolase ki kore glyceric acid to phosphoenol pyruvic acid ole convert kori diye, right? And lastly, we have this enzyme. Finally, pyruvic acid form koribole kuntu acid, enzyme help kore pyruvic acid kinase. So these are the 4 enzymes that we will read. Points bilak pisot likhim. Egita 4ta enzymes mon rakhiba step wise hodibo pare. Splitting enzyme ki, dehydrating enzyme ki, starting enzyme ki, and ultimately enzyme responsible for forming pyruvic acid. 4 enzymes people. Next, coming to ATP formation and ATP consumption, no? Glycolysis process, the first step of respiration, so obviously ATP to produce hoboi lakibo. So ATP keneke hoise either directly, remember we have used a word called as substrate level phosphorylation. Substrate level boli kote tumi gutei bilak step jot ATP formation dekha paisa in the form of ATP, that is what is called direct formation or substrate level. Kisuman step ki hoise NADH2 form hoise, NADH2 bhangi pela ako ki form koribo, ATP form koribo, kintu etu directly hua nai. Junto step directly hoise, he directly ami ki boli kom substrate level. Next is, if you remember a little later, what we have written is glycolysis or key synthesis hoy, ATP synthesis hoy or NADH2 synthesis. Ultimately, sobe ki form kore, ATP form kore. What is the total number of ATP that is formed? Ki likhisilu 8 ki ho karone soason, yate 2, right? Yate 2, right? 4 aru NADH2 kiman hoise? 2 molecules se kiman produce koribo? 2 molecules se kiman form? Eta molecule 3ta produce kore. 6 yate kiman hol? 7 8 tate kiman hol? 9 10, right? So if you are going by this, ami ATP production as of now ami kiman dekhisu 10 ta. Kintu ami ki koiso 8 ta. Why 8? Because 2 of them have been consumed in the process, right? So যেহেতু starting of the reaction ATP production নহয় কিন্তু ATP consumption process হয়, total যিমান ATP আছে তাৰ পৰা আমি 2 minus কৰি দিওঁ এইটো ষ্টেপত আৰু এইটো ষ্টেপত। So ultimately synthesis কিমান হয় 8 তাৰো 8 ৰ ভিতৰত ইয়াতে 2 তাতে 2 আৰু ইয়াতে 6 clear okay next is this word dehydrogenase word to monot rakhiba because this is a word or this is the name of an enzyme that we are going to study during Krebs cycle also, right? NADH to dehydrogenase, FADH to dehydrogenase. Jetia dehydrogenase word to dekha poa at that step what you have to remember dehydrogenase e hodai reducing cofactor produce kore. Reducing cofactor বুলি ক'লে আমি তিনটাৰ নাম জানো ন NADPH2 NADH2 আৰু FADH2 NADPH2 associated with photosynthesis NADH2 আৰু FADH2 associated with respiration so dehydrogenase jote থাকিব তাত কি ফৰ্ম কৰিব NADH আমি যেতে Krebs cycle draw কৰিম ন jote তুমি dehydrogenase enzyme to দেখা পোৱা তাতে দেখা পাবা যে either FADH2 gets synthesized or NADH2 gets synthesized so dehydrogenase enzyme is responsible for production of the reducing cofactor, right? Next point question ahise NADH2 kuntu step or produce korise you have to be precise plus precisely nukhudi pelai if you see etu ki ase so 2 phosphoglyceraldehyde right etu ki ase glyceric acid so aldehyde jete acidole convert hoise tetia he NADH2 produce hoise ba aldehyde jete acidole produce hoise tetia he NAD got reduced mane NADH2 production hoy no it's the same thing NAD was already present in the cell NAD H positive low pelai ki convert korile NADH2 form korile so NAD plus H tamane NAD ki hoise reduction hoise nai so question ahe reduction of NAD occurs at which step reduction of NAD occurs at precisely a2 di dile to bhalei jedi di die ki dibo reduction of NAD occurs at during conversion of aldehyde into acid right phosphoglyceraldehyde converted into phosphoglyceric acid aldehyde jete acidole convert hoise tetia ki produce korise NADH2 produce this is the way how we will read the some other points of importance from this part apart from the reaction to mukhasta korimi right so eta ami ketaman point likhu yar upor তলত এটা বস্তু লিখা ultimately glycolysis could be represented by মই ইয়াতে কন্টিনিউ কৰিবলৈ জেগা নাই এই কাৰণে মই বেকাকে লিখিছো Glycolysis gets converted, glucose gets converted to form pyruvic acid. Together with pyruvic acid, what they form ATP and NADH2, keta ATP, 2 ATP, and ultimately what they form is 2 NADH2. Our coat hoy cytoplasm. Cytoplasm is also called as cytosol. I think this is understood. এইটাটো বুজি পালো। pacemaker enzyme. So, phosphofructokinase code role to swa glycolysis. Phosphofructokinase ki ase fructose 6 phosphate into fructose 1,6 diphosphate, right? It is a deciding step. Tar pisote ki hobo je it split koribo, right? So, that's the reason. So, this is all about glycolysis. So, out of the 3 products that needs to get synthesized in complete oxidation, it is only ATP that gets synthesized in the first step, that is glycolysis. Second point, glycolysis is something that is common for both aerobic and anaerobic. Third point, where does it occur? It occurs in cytoplasm, right. If this is over, we will talk about the next step, that is Krebs cycle. এইখিনি বাই কিবা After glycolysis, what happens is pyruvic acid gets formed, kintu pyruvic acid needs to get further broken down, no? Akko breakdown kiyo hobo lage? Because the 3 products, as we have discussed, hey gutei 3ta product etialake dekha nai. It's only ATP that we can see, but carbon dioxide evolution or water evolution etialake hua nai. Because of that reason, pyruvic acid needs to further break down. And if you remember, in the last class ami okkoman eta idea disil, no? If you are talking about a cell like this, ekini jega ki boli kom ami cytoplasm. Cytoplasm or ki hoy ase, glucose gets converted to pyruvic acid, right. Next glycolysis or next step to ki Krebs cycle. Krebs cycle kot hoy mitochondria. So a pyruvic acid further breakdown kuntu step ot hobo? Krebs cycle ot hobo, kot hobo, mitochondria hobo. So the next connecting link should be obviously mitochondria jot niki ATP to synthesize hobo. So pyruvic acid jodi further breakdown hobo, first to further breakdown hoy pelai ki form koribo, further breakdown hoy pelai carbon dioxide aru water form koribo, carbon dioxide aru water form koribo ar tar logote akko ki form koribo, obviously ATP to form koribo, right. Next is second step Krebs cycle kot hobo lagibo, mitochondria hobo lagibo. Mitochondria bhitorot jodi Krebs cycle hobo lage, Krebs cycle start koribo lagile pyruvic acid yale humai ahibo lagibo, right. But we know that pyruvic acid is impermeable to mitochondrial membrane. Hey karone pyruvic acid ki hoy in the perimitochondrial space, a space to kami ki boli koiso perimitochondrial space, pyruvic acid gets broken down into acetyl coenzyme A, right. Kot hobo pyruvic perimitochondrial space. Perimitochondrial space or pyruvic acid bhangi jabo ki hole acetyl coenzyme A, right. Pyruvic acid to acetyl coenzyme A tak ami ki boli kom? Link reaction, right. So glycolysis aru Krebs cycle of link kune kore? Acetyl coenzyme A. Acetyl coenzyme A is a product of ki link reaction. Who gets converted to acetyl coenzyme A? Pyruvic acid gets converted to acetyl coenzyme A. Kot convert hoy in the perimitochondrial space. Tar pisot acetyl coenzyme A jot yat form koribo, ekhini jagat jodi form korise, it will enter inside the matrix of mitochondria and it will be taken in by oxaloacetic acid, the starting point of Krebs cycle, and then Krebs cycle will start. Pyruvic acid directly oxaloacetic acid accept koribo nware, it has to get broken down. Hey breakdown reaction to kami ki boli kom? Link reaction. Link reaction e ki kore? Glycolysis or Krebs cycle ok link kore. Clear? Kiba doubts ase niki? So first what we will do is we will write about the link reaction and then we go to Krebs cycle. Heading correction, link reaction. So glycolysis karone jonto pyruvic acid form korile, what happens to pyruvic acid? The pyruvic acid gets converted to acetyl coenzyme A. The reaction is called as link reaction. When pyruvic acid gets converted to acetyl coenzyme A, kot hoy ase perimitochondrial space. Now since this is also a reaction, kiba eta reaction hole kiba eta produce hobo lagibo. Jeneke glycolysis or glucose got converted to pyruvic acid, and what produced is only ATP in the form of ATP and NADH2. So etia ki hobo, pyruvic acid will get converted to acetyl coenzyme A. So reaction mazot tatu to kiba eta produce hobo lagibo. So could you guess the product ki produce hobo? Are you understanding the question? The question is glycolysis oto kisuman products produce hoy. Since link reaction is also a reaction oto kisuman product produce hobo. If you are talking about product, we are not talking about what is get converted to what. Glycolysis or glucose gets converted to pyruvic acid. Link reaction or keep convert hobo, pyruvic acid gets converted to acetyl coenzyme A. Kintu Synthesis কি হ'ব কিবাটো synthesis obviously হ'ব লাগিব glycolysis ত কি synthesis হৈছিল ATP আৰু NADH2 তো এইটোটো এটা ৰিঅ্যাকশন এই ৰিঅ্যাকশনটো কিবাটা synthesis হ'ব লাগিব তো কি synthesis হ'ব just try to guess একটো কি হয় link reaction is also a part of respiration each and every step of respiration will lead to the formation of ATP to produce hoboi right next is just look at this name pyruvate dehydrogenase complex আছে তো dehydrogenase complex আছে তাৰ মানে তাত কি ফৰ্ম কৰিব reducing cofactor produce কৰিবই তাৰ মানে NADH2 produce কৰিব NADH2 ultimately কি ফৰ্ম কৰিব ATP ফৰ্ম কৰিব right এটো ওয়ার্ড বুজি পালো next চোৱাচোন pyruvic acid কি হলে কনভার্ট হৈ যায় acetyl coenzyme A process to কেনেকে হয় oxidatively decarboxylated মানে carbon dioxide evolve হ'ব নে use হ'ব carboxylation মানে কি use of carbon dioxide no carboxylation reaction first step of that reaction we have read about no ইয়াতে কি হৈছে decarboxylation process হৈছে so decarboxylation process মানে pyruvic acid acetyl coenzyme A লে যেতিয়া কনভার্ট হ'ব তাত কি হ'ব decarboxylation হ'ব মানে carbon dioxide evolve হ'ব নহয় হ'ব so we have an idea so কি হ'ব within this reaction carbon dioxide evolve হ'ব আৰু reducing cofactor produce কৰিব reducing factor এ কি ফৰ্ম কৰিব ultimately ATP synthesis কৰিব clear so we'll start the link reaction with pyruvic acid কিমান কার্বন 3 কার্বন কি ফর্ম করব acetyl coenzyme A acetyl coenzyme A কিমান কার্বন হ'ব obviously process to কি হ'ব decarboxylation মানে এটা কার্বন গুছি যাব তো acetyl coenzyme A কি হ'ব 2 কার্বন compound so oxidatively কি হৈছে decarboxylated হৈছে plus dehydrogenase enzyme আছে হে কাৰণে reducing factor produce কৰিছে NADH2 এতিয়া এখিনি পাৰ্টৰ পৰা অকমান কোৱেশ্চন আহে কোৱেশ্চন কেনেকৈ আহে কোৱেশ্চনটো you have to read it properly first question is related to what are the things that get synthesized ATP produce হয় নে নহয় obviously হয় substrate level হয় নে via ETS হয় via ETS. Substrate level নহয় কাৰণ আমি directly ATP দেখাই নাই ইয়াতে, right? So via ETS হলে কিমানটা ATP produce হ'ব হেতু যোৱাৰ আগতে first let's talk about carbon dioxide and NADH2. Question ahe, what is the number of carbon dioxide and NADH2 produced during link reaction? 1 dekhisu kintu you have to read the option carefully. Right, question to bhalke poribo laibo kihor karone 1 pyruvic acid will form 1 acetyl coenzyme, 1 carbon dioxide, 1 NADH2 kintu pyruvic acid keta form korisil duta to duta hole etu cycle dubar hobo nohoy so dubar hole duta carbon dioxide duta NADH2 produce hobo nohoy. So question to jodi ahe glycolysis ot acetyl coenzyme A form kora loke kiman carbon dioxide or kiman NADH2 produce hoy, then you have to make it double because we know glycolysis word to thakile we are talking about the formation of 2 pyruvic acid. So 2 pyruvic acid will lead to the formation of 2 carbon dioxide and 2 NADH2. Kintu as of now jodi ami 1 pyruvic acid to 1 acetyl coenzyme A hoy carbon dioxide kiman produce korile 1 NADH2 kiman produce korile 1, right? 1 NADH2 is equal to kiman 3 ATP. Aru jodi khude up till the formation of acetyl coenzyme A, what is the number of ATP that has been synthesized? Tetia noy tetia ami ki koribo laibo glycolysis khini aru plus yate, right? Karon question to ki ase glycolysis or pradise glycolysis word to na thaki pelai jodi okol link reaction die tetia hole ami etu calculation koribo laibo je tat 6 hobo tetia কারণ দুটা পাইরুভিক অ্যাসিড দুটা অ্যাসিটাইল ফর্ম করিলে দুটা অ্যাসিটাইল যেটা ফর্ম করিলে এটা ডাবল ডাবল হয়ে যাব এটো যদি আমি দুটা পাইরুভিক অ্যাসিড দিও এটো হলে দুটা হয়ে যাব এটো সব ডাবল হয়ে যাব রাইট সো অপশনত কোনটো আছে এইটো বস্তু ভালকে চাম আৰু যদি কোৱেশ্চনটো গ্লাইকোলাইসিস ওয়ার্ডটো আছে তেতিয়ালে একদম স্টার্টিংৰ পৰা এতিয়ালকে গুটেই ক্যালকুলেট কৰিব লাগিব ক্লিয়াৰ so this is the link reaction next is the second step, that is Krebs cycle. Heading koriba Krebs cycle. It is also called a citric acid cycle because the first product that is formed is called is citric acid. গ্লাইকোলাইসিছৰ নেক্সট ষ্টেপ কি ক্ৰেব চাইকেল ক'ত হ'ব মাইটকণ্ড্ৰিয়াৰ ভিতৰত ক্ৰেব চাইকেল কেইটা চাইকেল হ'ব দুটা চাইকেল কিহৰ কাৰণে দুটা পাইৰুভিক এচিড ফৰ্ম কৰিছিলে ৰাইট সো এচিটাইল ক্ৰেব চাইকেল আৰম্ভ কোনে কৰিব এন্ট্ৰেন্ট মলিকিউলে কৰিব one that comes in no who will come in obviously acetyl coenzyme তো acetyl coenzyme কেইটা প্ৰডিউস কৰিছিল দুটা কাৰণ পাইৰুভিক এচিড দুটা ফৰ্ম কৰিছিল সো কোৱেশ্চন আহে এটা গ্লুকোজ মলিকিউল কমপ্লিটলি ব্ৰেক ডাউন কৰিব লাগিলে Krebs cycle কেইবাৰ ৰান কৰিব লাগিব দুটা 2 rounds of Krebs cycle per glucose molecule okay next আকৌ লিখিব লাগিব Amphibolic pathway we understand catabolic anabolic দুইটা যদি হৈছে তেতিয়া তাক আমি কি বুলি কম amphibolic Why it occurs in the matrix of mitochondria? Because Krebs cycle is also an enzyme oriented reaction, right? Mediated reaction are all the enzymes except one is present in the matrix of mitochondria. I think this is something that we have learned, no? Mitochondria jeta porisilu, mitochondria sob enzymes bila Krebs cycle karone jimanbur enzymes lage kot ase matrix ot ase. Kintu eta enzyme to be precise, succinyl coenzyme বা সাকসিনিক অ্যাসিড যেতিয়া কনভার্ট হৈছিল তেতিয়া হলে হে এনজাইমটো কোড আছে ইনার মাইটোকন্ড্রিয়াল মেমব্রেনত আছিল দিস ইজ ওভার Next we'll continue. Egita holle gute egita hol. entrant molecule of Krebs cycle, acceptor molecule of Krebs cycle. Entrant molecule acetyl coenzyme A সোমাই পেলাই কোনোবাইটো accept কৰিব ন so mitochondria ভিতৰত কি আছে oxaloacetic acid so acceptor molecule কি oxaloacetic acid oxaloacetic acid কিমান কাৰ্বন 4 কাৰ্বন acetyl coenzyme A কিমান কাৰ্বন 2 কাৰ্বন ultimately কিমান কাৰ্বন 6 কাৰ্বন citric acid কিমান কাৰ্বন 6 কাৰ্বন compound right Amphibolic কিহৰ কাৰণে কয় because catabolic reaction আৰু anabolic reaction সব হয় Krebs cycle কাৰণে দুটামান বিৰাট important product synthesis হয় I'm not talk about what gets evolved right এটাৰ পৰা এটালে convert হয় ন বস্তু so কি বস্তু important মই এটো লিখি মেখিনি শেষ কৰা এটো লাইনটো হৈছে ওকে লাষ্টটো লিখি দিয়া ইয়াৰ পিছত কন্টিনিউ কৰিবা হা এগিটা থৈ দিয়া এখিনি লকেটো আমি অলৰেডি লিখিলো এখিনি হৈ যোৱাৰ পিছত ইয়াৰ পিছত নেক্সট ইয়াৰ পিছত কন্টিনিউ কৰিবা হা এটো পইণ্ট তাৰ পিছত নেক্সট এটো পইণ্ট এখিনি নুমুছাৰ কিবা এটা ৰিজন আছে উইল ডিসকাস লেটাৰ Last line over. Okay. So as we have written now, a Krebs cycle or the citric acid cycle is called as amphibolic pathway because there are certain important products that are formed. Out of them, one is succinyl coenzyme A. If you see Krebs cycle or if you remember Krebs cycle, no, citric acid, oxalocitric acid, then they convert hoy eta step out ki hoise. So succinyl coenzyme A hoise. Alpha keto ase, apsa succinyl coenzyme A ase. right? So succinyl coenzyme A is very important. Why it's important? For the synthesis of porphyrin ring compound. Talking about porphyrin ring, we have already discussed the structure of the chlorophyll. Structure of chlorophyll মনত আছে নে? It had a head and it had a tail. Head tut ki asile? Saita pyrrole ring asile nasile? Something like this, chlorophyll, right? Yate ki asile? Mg, yate saita ring asile, right? এইটো পাৰ্টক কি বুলি কৈছে porphyrin head এটো কি কৈছিলে phytol tail right so এনেকা porphyrin ring থাকা কিছুমান compounds আছে porphyrin head মানে কি চাৰিটা কি থাকিব pyrrole ring থাকিব open tetrapyrrole structure closed tetrapyrrole structure এখিনি আমি already পঢ়িছো so porphyrin head or porphyrin ring compounds এতিয়ালৈকে আমি কি পঢ়িছিলো chlorophyll but now we know it is not only chlorophyll but we have certain examples also so question keneke ahe identify the porphyrin ring compounds or identify the porphyrin which among them is a porphyrin ring compound chlorophyll is something that's common that we have already read about it apart from chlorophyll ihotoru ki thake eta porphyrin ring compound thake a porphyrin ring ka compounds bilak kone synthesize kore succinyl coenzyme A if you remember from the chlorophyll point of view we have studied we have talked something called as precursor of chlorophyll jedi monot ase biosynthesis of chlorophyll ami korisilu biosynthesis ami ki likhise succinyl coenzyme A plus glycine will give you key protochlorophyll will get converted to key chlorophyll in presence of light so ultimately chlorophyll kune form korise succinyl coenzyme code form korise Krebs cycle right so Krebs cycle kisuman dutaman products especially succinyl coenzyme A aru alpha ketoglutarate acid ei duta birat important for other point of view right so succinyl coenzyme A importance ki porphyrin ring compound form kore etia likhim alpha keto Amphibolic pathway that important product duta ase eta hol succinyl coenzyme A eta hol alpha ketoglutarate acid ami olop pisot cycle to akim so we'll be talking about this too succinyl coenzyme A is associated with forming porphyrin ring compounds alpha ketoglutarate acid is responsible in the formation of a amino acid what is the name of the amino acid glutamate or glutamic acid nitrogen metabolism ami korisu niki pasot nitrogen fixation korisu niki nitrogen fixation অকমান কৰিছিলো নেকি if suppose we are talking about cyanobacteria যেটা কৰিছিলো biological classification অকমান আমি nitrogen fixation কৰিছো নেকি as far as the process is concerned না মনত নাই বাৰু ঠিক আছে plant growth and hormones আমি details কৰিম right plant growth and development is the last chapter so we'll be talking about it in details as of now so nitrogen fixation বা nitrogen metabolism or logot ki associated hoise glutamate or glutamic acid nitrogen fixation mane ki je atmosphere or nitrogen to kot ahibo soil mix hobo soil or pora plants e loi lobo right jetia plants e soil or pora nitrogen loi they accept it in the form of N2 ne something else N2 so plants e nitrogen loi ne no loi obviously loi korpra lobo nitrogen kot thake atmosphere nitrogen ki form thake N2 form to plants e N2 form directly nitrogen lobo pare ne noare so it has to get fixed matlab mane ki je soil ot ahi pele bele compounds le convert hobole ibo ki hole convert hoy nitrite or nitrates hole convert hoy so N2 gets converted into NO2 and NO2 gets converted into NO3 ultimately NO3 form plants bilake absorb kore right so plant soil ot ki ase NO3 NO3 কোলে যায় প্ল্যান্টৰ ভিতৰলৈ যায় প্ল্যান্টৰ ভিতৰত NO3 what happens to NO3 NO3 gets converted to NO2 NO2 gets converted to ammonia ammonia gets converted to amino acid তাৰ মানে ultimately atmospheric nitrogen is present in which form inside the plant body in the form of amino acid plants as a nitrogen ললে nitrogen লৈ পেলাই কি কৰিলে কিয় লাগে নাইট্ৰোজেন আমি এটা চাউল কিনি আনো কিহৰ কাৰণে ভাত বনাবলৈ right plants and nitrogen lole nitrogen লৈ পেলাই কি কৰিব কিবা এটা তো কামত আহিব লাগিব ন so nitrogen কিহৰ কাৰণে লাগে কাৰণ বিৰাট all the important compounds সব বস্তু ফৰ্মেশনৰ কাৰণে nitrogen লাগে আৰু plants or হে nitrogen to directly soil পৰা লব নোৱাৰে হে কাৰণে with the help of microorganisms nitrogen fixation process হয় আৰু তাৰ পিছত they take it in the form of NO3 but this NO3 needs to get converted to usable form inside the plant body নাইট্ৰোজেন তো ললে নহব ন ইউজ তো কৰিব লাগিব চাউল তো কিনি আনিলেই হ'ব নেকি ৰান্ধিবওতো লাগিব right so plants কি কৰে নাইট্ৰোজেন তো লৈ ললে কি ফৰ্মত NO3 ফৰ্মত so inside the cell obviously NO3 is present but they cannot use it to use NO3 needs to get converted again কি হলে NO2 লে কনভার্ট কৰে NO2 কি হলে কনভার্ট কৰে অ্যামোনিয়া লে কনভার্ট কৰে অ্যামোনিয়া কি কৰে সেলটোত থাকা বেলেগ বেলেগ কম্পাউন্ডছৰ লগত কম্বাইন কৰি পেলাই অ্যামিনো এসিড ফৰ্ম কৰে amino acids bilakor we know that amino acids 20 type of amino acids itur lokot itu combine kori kori beleg beleg amino acids form koribo pare what is the first form amino acid inside the plant it's glutamic or glutamate right so ultimately moi jonu question nitrate will get converted to ki NO2 inside the plant cell NO2 ki hole convert koribo ammonia le ammonia plus alpha ketogluteric acid combine together to form glutamic acid নাইট্ৰোজেন কি হলে কনভার্ট হল অ্যামিনো অ্যাসিড হলে কনভার্ট হ'ব লাগিলে অকল নাইট্ৰোজেন ইন দা ফৰ্ম অফ NO3 ডাইৰেক্টলি অ্যামিনো অ্যাসিড হলে কনভার্ট হৈ যাব নোৱাৰে কাবাৰ লগত লগ লাগিব তেতিয়াহে অ্যামিনো অ্যাসিড ফৰ্ম কৰিব right so ammonia কাৰ লগত লগ লাগিলে বেলেগ বেলেগ বস্তুৰ লগত লগ লাগিব পাৰিব যদি আলফা কিটোৰ লগত কম্বাইন কৰি ৰিঅ্যাকশন টু প্ৰডিউস কৰে তেতিয়া তাহাতে কি ফৰ্ম কৰিব গ্লুটামেট so ultimately in one way what we can say is atmosphere of nitrogen ahile ultimately inside the plant body in the form of what is it available it is available in the form of amino acids aru tatkeo jodi precisely ami porhu amino acid is supposed to be very unstable because we know eta amino acid belegor logot combine kori beleg eta amino acid form koribo pare so it's not a stable form so we have to talk about the stable form amino acid e ki form kore amides form kore amides etia form kore glutamine amino acid tur naam ki glutamic acid right stable form to ki amide tur naam ki glutamine so ultimately atmospheric nitrogen is present inside the plant body in the form of amides what is the first form amide ki glutamine right so ultimately prothomote ki form koribo laibo glutamic acid form koribo laibo tar pisothe amide form koribo glutamic acid kude form koribo nitrate ammonia asile ammonia kar logot log lagibo alpha keto logot log lagibo ki form koribo glutamate so this is something that's related with nitrogen metabolism Krebs cycle juntu eta intermediate product that is alpha ketogluteric acid plays an important role in nitrogen metabolism clear okay etia aru eta sentence likhu that this was the anabolic steps involved with Krebs cycle next point Last point is something we have already discussed. Enzymes বিলাক ক'ত থাকিব matrix of mitochondria TCA cycle লগত associated যিমানবোৰ enzymes আছে বা citric acid cycle লগত যিমান associated enzymes আছে ক'ত থাকিব matrix of mitochondria except one enzyme. What is that one enzyme? Succinic dehydrogenase enzyme, which is also called as mitochondrial marker enzyme. Question আহিব পাৰে, identify the mitochondrial marker enzyme. Marker enzyme বুলি ক'লে কাক কয়? Succinic dehydrogenase ক'ত থাকিব? Inner mitochondrial membrane থাকিব. বাকিবিলাক enzymes ক'ত থাকিব? Matrix of mitochondria থাকিব. Right? This is understood. Just come to this point. So, as far as the number of NADH2, FADH2 and ATP is concerned, আমি তিনটা information লিখিছো. চোৱাচোন ভালকে গোটেই তিনটা পইণ্টত ন this point গোটেইকেইটা মাৰ্ক কৰি থবা হা এইটো এইটো আৰু এইটো সতে গোটেইকেইটা নাম্বাৰবোৰ বেলেগ বেলেগ হয় নহয় হয়নে সো ক্ৰেব সাইকেলটো আমি এতিয়ালৈকে ড্ৰ কৰা নাই হা এটা গ্লুকোজ মলিকিউল ভাঙিব লাগিলে কমপ্লিটলি যদি ভাঙিব লাগে ক্ৰেব সাইকেল হ'ব লাগিব ক্ৰেব সাইকেল কেইবাৰ হ'ব লাগিব দুবাৰ সো টিসিএ সাইকেল কেইবাৰ হ'ব দুবাৰ the first point is এবাৰ টিসিএ সাইকেল হলে কি বস্তু সিনথেসাইজড হয় তাত কি ফর্ম হ'ব NADH2 ফর্ম হ'ব FADH2 ফর্ম হ'ব আৰু কার্বন ডাইঅক্সাইড ফর্ম হ'ব আৰু ATP তো obviously ফর্ম হ'ব because it's a step of respiration right as far as ATP or energy is concerned energy in the form of ATP or GTP if you see in the cycle it is just one glycolysis of jeneke ami ATP kiman dekhisilu 4 dekhisilu properly produce kora minus kori pelai 2 hoisile kintu produce kiman korisile 4 so Krebs cycle in the form of ATP kiman produce koribo okal 1 ataku ATP form ot nahi pelai GTP form ot ahibo kintu we can consider both of them as same okal sugar to beleg kintu it will give you energy only guanosine triphosphate by adenosine triphosphate right so ATP buliu question ahibo pare GTP buliu question ahibo pare ekdom precisely jodi porha glycolysis ot ki synthesis hoy ATP Krebs cycle ot ki form kore GTP and ATP kune form koribo NADH2 or FADH2 okay now just see 4 NADH2 1 FADH2 and 1 ATP are formed from question 2 keneke ahe hetur upor question ahibo laibo yate tinta information the first information one round of Krebs cycle e kiman NADH2 FADH2 GTP produce kore second question two rounds of Krebs cycle e NADH2 FADH2 GTP kiman form koribo third question pyruvic acid e NADH2 FADH2 GTP kiman form koribo whenever we talk about pyruvic acid no jedi question to pyruvic acid ase tete hale ami link reaction or NADH2 consider koribo libo link reaction or pyruvic acid ki hole convert hosile acetyl pyruvic acid er pora acetyl coenzyme e convert hote keita NADH2 form korisil eta two rounds buli to likhi dia nai one pyruvic acid he koise so one pyruvic acid or কিমান produce কৰিছিল 1 NADH2 ঠিক আছে হেতু কনসিডার কৰিম 1 TCA cycle কি produce কৰে যদি NCERT বুকখন আছে if you want to see just see যেটা cycle to দিয়া আছে NADH2 কেইবাৰ produce কৰিছে চোৱাচোন আমি next class ত হেতু আকিম so we'll be talking about that class কেইটা form আছে NADH2 তিনটা FADH2 কেইটা produce হৈছে এটা right can you see that তুমি দেখা নাই ন Oh, usarle agi pele ekhon sail ban hole Krebs cycle swa tumi ki sai asa Krebs cycle ot swa NADH2 keita produce hoise tinta hoise na 3 steps ot FADH2 keita produce hoise eta ar GTP keita produce hoise eta hoy so one round of TCA cycle buli jodi precisely mention kore tetale ami ki likhim 3 NADH2 1 FADH2 1 GTP hoy ami hetu diagram to dekha paisu so total kiman hobo 9 1 FADH2 is equal to 2 আৰু 1 GTP ultimately 12 tane one round ot keta form kore 12 ta Krebs cycle kebar hoy dubar to two rounds ot kiman form korbo 24 ta clear okay etu bad dia so one round of TCA cycle etu two rounds of TCA cycle here double hoy jabo hol one pyruvic acid to aru Krebs cycle link hoy ase ne nai You cannot be separating pyruvic acid as a separate process and Krebs cycle as a separate process because there is a link reaction. So, if the question is asked about starting point to pyruvic acid hisabe loise ba 1 pyruvic acid ba 2 pyruvic acid koise tetia ami ki koribo lagibo je link reaction or eta NADH2 aru yare tinta NADH2 that will make you 4 NADH2. Baki bilak eke no 1 FADH2 1 FADH2 1 ATP 1 ATP কিন্তু 1 pyruvic acid আৰু 1 TCA cycle or eta point of difference 4টা NADH2 3টা NADH2 pyruvic acid বুলি question to আহিলে link reaction or NADH2 আমি calculate কৰিব লাগিব যদি question to আহে what is the number of NADH2 produced from 1 pyruvic acid ক'ত form কৰিছে glycolysis pyruvic acid or pisot ki hoise link reaction hoise Krebs cycle hoise so pyruvic acid e NADH2 keita produce korise eta produce korise pyruvic acid acetyl ole hote tar pisot produce korise tinta acetyl ahi pale Krebs cycle run korute so ultimately keita form korile 4ta right so jedi pyruvic acid or calculation to question ahe tetale ami link reaction to include koribo lagibo jedi pyruvic acid nahi pale directly 1 TCA cycle TCA cycle বুলি কলে আমি pyruvic acid consider নকৰো pyruvic acid বুলি কলে কিন্তু TCA টো আমি consider কৰো is it clear TCA cycle বুলি যদি specifically mention কৰি দিয়ে TCA cycle is Krebs cycle so we are concerned with only the products of the Krebs cycle citric acid form কৰিলে তাৰপিছত ultimately আকৌ oxaloacetic acid হল সেই cycle টোত কি produce হৈছে তিনটা কিন্তু যদি আমি pyruvic acid কও link reaction to মাজত আছে ন so হে কাৰণে link reaction or product to আমি calculate কৰিব লাগিব so that makes the calculation as to 4 NADH2 link reaction to FADH2 produce one asile ATP produce one asile ki produce is NADH2 so yate tinta tat eta next point is 1 pyruvic acid na 2 pyruvic acid we know 2 pyruvic acid so 1 pyruvic acid or the product hegita 2 pyruvic acid or obviously product egita hobo so question kita sabole gole it's the same thing but you have to read the question properly ki question khudise 1 pyruvic acid khudise na 2 pyruvic acid khudise 1 TCA cycle khudise na 2 CCA cycle korise ne pyruvic acid buli khudise na TCA cycle buli khudise as far as NADH2 calculation is concerned right so 8 point clear hoba I think this is all for today, right? Next class of Krebs cycle will draw. And if you want to draw it from home, the cycle to draw kori loi anibo para. Ar nohon class to ake dim. Thank you.